Oops, I just said something wrong. Oh, there we go. Hello, can you hear me? Yeah? Yay!
Yes.
Okay, good. Got another minute here. Let a few more folks join. Thank you. All right, well, it's about 7:30, so thank you guys all for logging in. It's nice to see some of you and interact with you in a different way other than just grading. I'm open to answering questions. I am recording it just so that you know. You can either send me questions via the chat feature, or you can just speak out loud if your microphone is hooked up and working. If anything comes through the chat, I'll make sure that I read that back to you guys. I think you can see it as well, but just in case, I'll read it back to you. We have about 6 people on the line here, so if you've got any questions, feel free to just jump right in.
Hi, this is Suzanne. Can you hear me?
Yep. Hi, Suzanne.
Hi, how are you?
Good.
I had a quick question. This has to do with the cyclic AMP inhibition of the myosin light chain kinase.
Mhm.
In the, I think it was the question where you were describing the relaxation of the beta-2 receptors for bronchial when we have a beta-2 agonist.
Mhm.
Could you just talk about that receptor chain of events real quickly?
Yeah, sure. Let me see. I can pull up. That's not what I want to do. Is this? Yes. All right. Want to take a look at one of my. I have the assignments pulled up, the answer keys pulled up, but it's
Okay. It was homework number 1.
there we go. Oh, that's 2. Of course, I want 1. There we go. All right. This is I've used Zoom before, but this is kind of the most I've used it with multiple people and sharing my screen. So
Okay.
let's scroll down to where that was.
Okay, that one. Yeah, that one right there.
Yeah. Okay. So beta-2 adrenergic receptors coupling to G-alpha S.
Right.
When activated, will activate adenyl cyclase, which causes the conversion of ATP to cyclic AMP.
Right.
And then when we get Oh, I see where you're coming from here. Okay, yeah.
Yeah.
There does seem to be a skip step. So once you get the increased concentrations of cyclic AMP, that will result in, think about it, and you can correct me if I'm wrong because I haven't thought about it in a little while. It should result in reduced concentrations of calcium. And then if you have less calcium available, the kinase won't work. Let me see if I can pull up a different picture. Oops.
And I'm trying to think about it in terms of, okay, it's the beta 2 receptor. You know, we're not looking at a muscarinic receptor.
Mm-hmm. Sometimes this is like my favorite way of looking things up. Oh, actually, you know what? To just look for a picture. So it looks like the calcium levels actually decrease, but there is a direct tie between calcium levels decreasing. I'll make this nice and big. So if we look here, if we were to have GS activation, have increased levels of cyclic AMPO, and this one just goes you directly to inhibiting the myosin light chain kinase. So it must be the matter of having increased concentrations of cyclic AMP resulting in the inhibition of the kinase. It could be that you're removing levels of ATP, and so since you have less energy in the cell, the kinase isn't going to be able to transfer the phosphate, that one phosphate group. It could also be, and I guess on this picture they're not showing this, but I always think about it too in terms of increasing concentrations of calcium. And so the effect that calcium has on the light chain kinase. Although in this image, they're showing that going through the GQ coupled receptor and not the GS.
And I'm sorry, what are you looking at?
Oh, can you still see my screen?
Oh, we're still seeing the homework response.
Oh, good. Oh my gosh, I'm like totally explaining this to you and you're not. That would be pretty confusing. Let's see. There we go.
Okay.
Yeah.
All right.
Let's see if I can move this. Yeah, so in this picture here, so they show the GS coupled receptor being activated, increasing cyclic AMP concentrations, and they show that as having a direct effect on the
Okay.
myosin light kinase. And like I was saying, I think it could be maybe because you're taking away ATP because the ATP is getting converted into cyclic AMP. I'm not sure if that's like a direct correlation though. You probably have higher concentrations of ATP in the cell than you do of cyclic AMP always, just at resting. And then I always think about the myosin light chain kinase being inhibited because of the interaction with calcium, but they're showing that that's regulated by a GQ coupled receptor and not the GS. So, I mean, I guess it kind of it's not in direct alignment with the way that I think about these things, but I guess in this image here, you can see that the cyclic AMP levels directly inhibit myosin light chain kinase. I'm trying to look to see.
Well, I mean, it looks like that if it does directly inhibit it, then it's preventing something between the MLC going to the contraction. So it looks like that if it's inhibiting the light chain kinase, it's preventing that relaxation reaction going to the right. So I mean, I can memorize that, I guess, but
I do want to clarify is that so if CAMP is built up, the calcium decreases, correct?
I'm thinking. Hi, Holly.
Hi.
Let me see if I can find another one. It has to deal with the myosin light chain kinases phosphorylating the myosin heads. This actually might be a good website that I can share with you guys if you wanted to just look up CV physiology.
yeah.
I think I misspoke earlier when I said there was decreases in calcium. I think there's actually increases in calcium levels. And
Would it be because the CAMP, as it increases, it interferes with the kinase, and because the kinase is being inhibited, it looks like it's building up in calcium, so the calcium's not being used? Is that what it is?
it could be that. It could also be, and I want to confirm this before I say this to you guys. The beta gamma subunits will also have an effect on channels, and it could be that they are opening. Oops. I'm not sure which one you guys are looking at now. Okay, so you're looking at this. So the beta gamma subunits will also kind of change some of these channels, and I believe they also activate calcium channels. So if we were to look at this picture again, seeing the activation of these calcium channels opening up and bringing calcium in, calcium levels increase. I'm just reading through this paragraph next to it. If you guys can you see what I'm highlighting? So it says the mechanism by which intracellular calcium increases either from increased influx from calcium through the cell through calcium channels, or by the release of calcium from internal stores like the sarcoplasmic reticulum. Free calcium will bind calcium commodulin, activates the kinase, the enzyme responsible for phosphorylating the light chain.
I think we're still looking at the diagrams.
From the internet.
Yeah, I don't see what you're reading.
All right, well, let me show you the website, or actually, you know what? I'll send this link in the website in the chat, and this will actually get. I'm saving the chat as well, so.
Well, I know with the homework number 3 on the back, she had those pictures that you could follow. You know, so to see like how calcium comes in and causes a contraction and stuff. So
Okay, so does that picture actually help explain then where the increase in cyclic AMP leads to the relaxation of the bronchodilation, the bronchioles? Oh, here it is. Okay.
Okay, I'm looking for cyclic AMP. Yeah, and then there's a top one right there that shows that there's calcium coming in, and then ATP, beta 2 agonist, CAMP plus, contraction. So that one's this one right here.
It's the top one, yeah.
You see, yeah. So
Yeah.
So if that I don't know if that might help. I just wanted to point that out.
Yeah, it does. Okay, so it's an alternative pathway.
Yeah.
Is that you, Kristen?
Yeah.
Where was that one? Can you repeat where that was?
Yeah. So on the homework, on the module 3 homework, and then I don't know if y'all are familiar with that. And then on the back, she had graphs that she wanted us to go over to show the different how it comes in, and there's a graph at the bottom right there.
Oh, okay. Did these go along with her videos?
Yeah, they go along with her videos. She said to make sure we're familiar with these, how calcium comes and causes contractions in the muscles. So that was an overview to show you how it does that.
So the increase in cyclic AMP takes you from the myosin light chain kinase to the myosin light chain kinase phosphate, and then from there, I guess the cyclic GMP comes in and does. Do you see why I'm saying I just don't? It's not connecting.
Yeah, we're from cyclic AMP to calcium, right? That's where we're.
Well, maybe not. I mean, it just, I don't know. The homework, the answer in the homework was hard for me to apply to this. So, go back up to that homework. Let's see, the homework answer.
I just know from the diagram it shows calcium coming in, and then it binds to calmodulin, and then it's going to cause a contraction, which you have potassium going out of the cell, and then you have ATP, which is causes a rise in CAMP, and which is going to cause it's going to interact with the MLCK, and then you're still going to have your rise in GMP, and then it's going to, depending on where it interacts at the bottom, you're going to have relaxation or not. So that might help.
Yeah, and it does, but I'm saying going from the light chain, was it the light chain CK, and then it's some kind of phosphate group, and then your cyclic GMP comes in and inhibits that to cause the relaxation. So I guess the flow goes. Do you see? There's no arrow between the MLCK PO42.
Mm-hmm.
Just your GMC, your cyclic GMP is just coming in with that. I guess that's the answer.
Oh, so I'm looking at it now too in that diagram. Give me 1 second to kind of follow along. Phosphorylated. Okay, so it might be, and let me see if I have the diagram pulled up that you guys are probably all looking at. This one. So here.
Oh, I see.
So here, what they're indicating in this picture is that the kinase, the enzyme itself, has been phosphorylated. And what I would say then is that it's not functioning. So if it's not working, then it can't, it'll cause relaxation instead of contraction. So here, I think the difference here is that they're saying that the actual enzyme itself is phosphorylated, not that the substrate is being phosphorylated, which is happening over here on the contraction side. And then this, another good point that Suzanne, you were talking about, this is cyclic GMP and not AMP down here. So this is kind of like a different signaling pathway, which often are used sometimes I use them interchangeably, but you guys remember from the third journal article, we were talking about cyclic GMP and not AMP. So it's just a slightly different signaling molecule than our cyclic AMP up here. Does that make sense? A little bit more sense? So if you have increased levels of cyclic AMP from our beta-2 stimulation, then the myosin light chain kinase, the enzyme itself, will be phosphorylated and turned off.
And that in turn will, because it's no longer being used with continuing on, then the calcium will be built up.
So if that myosin light chain kinase is not going forward to cause the contraction, then by default you're going to have relaxation.
Yes. Yeah. Okay. Thank you for asking that, because I haven't looked at that in a really long time.
I have to think about that one.
Yeah. Get out of this.
I haven't checked my posts tonight. So have you talked to us about the format of the midterm in terms of?
No, I haven't. Oh, there. Wait. Let me turn off the sharing if I can. I don't know how to do that. Whatever. I'll just keep talking. So I haven't. Basically, if you open up the midterm, if you click on that link, there will be directions. And so that gives you a little bit of a description, but really it just is a lot of kind of multiple choice mixed in with some open response. There are some essay questions that are somewhere between, 5 points maybe. The total exam is 130 points, I believe, which you'll see if you click on that. So you can click on that link and not start the exam yet. If you want to like get there and see the instructions, there will be like a thing that you actually click that says start the quiz. And then you'll go through it. What else? I will say last year when people took the exam, they said that it was really helpful to not just go in chronological order, but to actually scroll through and answer everything that they knew first, and to answer the questions that were worth more points up front, because the time is crunched. So one of the things that we do is we make the exams a little bit longer for the time period, so that we ensure that people aren't looking up answers on the internet. And so what that means is it is kind of you're kind of crunched for time. But it's also possible to move through the exam and answer it all. So it's just that you got to kind of keep moving.
Okay. That's helpful.
Yeah, that was super helpful. And I didn't have that last year for the midterm. But then once I got that feedback from the midterm, I was able to tell students for the final. And at the final, they were like, oh, that was so much better to know that we can scroll through and find which ones we know first. So, and you'll see too, I have, it's broken down by module. So it'll be like module 1. And then in each of the modules, there are questions from the journal articles in there. Yeah, so you're going to want those. But if you have the journal articles in front of you, I don't think you should not spend time going back and memorizing those articles. If you just have those assignments in front of you, you'll be able to answer the questions that are coming up. And so for those, I also I screenshotted some of the figures and I just said, what did this figure tell us? And then it's multiple choice. So if you can just kind of see the figure, be like, oh yeah, that's what that is, and read through the answer choices, you'll be fine.
Okay.
The midterm, like the homework 3, which was like a practice midterm, that's very similar to, it's just longer, but that's very similar to the style of questions that are on the midterm.
And are we, for example, how detailed do we need to be in terms of knowing which ions come in at what time to cause the depolarization or the repolarization? Or do we need to know that, or can we just, or is the concept good enough?
I'm trying to think. So that would be like from the more recent stuff, like the cardiovascular, or no, the neuro stuff.
Yeah, I'm just thinking like, there wasn't a whole lot of focus on the depolarization of the acetylcholine and that the receptor there. You know that the sodium comes in and the action potential comes along, that kind of thing. Mm-hmm. How detailed do we need to be for that?
I think as long as you're in line with what's in the answer keys for the homeworks, that's fine. That's all I'm looking for.
Okay.
And I want to make sure the same is true for Dr. Rometz's stuff. Dr. Rometz's stuff, I would strongly recommend having the homework that she had for you guys. I think it was homework 2 and homework 2. Having that in front of you, she does want you guys to know specific drugs. Mm-hmm. So it's just kind of a difference in how we teach. She likes you guys to know specific drugs. I'm more like, if you just know the drug class, then you kind of understand what's going to go on. So, but the first page, or maybe it was the second page where she has all the drug names, having that in front of you, I think will be helpful for the exam. Okay. And obviously it's allowed. There's no way we can regulate that. So. How do I stop sharing this?
I think you have a question in the chat section.
Oh, good. I'll go in order. So in the lecture material regarding pathophysiology for cholinergic medications, there is a question about an overactive bladder being better or worse with the use of a cholinergic medication. My question is, would it be better to use an anticholinergic drug for this scenario? Would a cholinergic medication overstimulate an overactive bladder? So, does anybody have any thoughts on that? I can pull up the kind of the answer key. But does anybody have any thoughts on that before I go ahead and do that?
I would say anti. That's just my guess.
I mean, if you've got an overactive bladder, your body is making you pee, and you don't want to pee, so you want to be anti-cholinergic.
All I had to say is M3 makes you pee.
That was a great hint, though. It does help you think about it.
Yeah, so you would definitely want to go anticholinergic there for an overactive bladder. And that's what we do use, right? The pharmacist in the room, right? We use oxybutyn.
Yes.
Yeah. I'm not a pharmacist.
Yes, Japan. and all the ones you see on the commercials with the pipes and the flow and the vesica. Yeah, Karen, yes.
All right. Another question is, we get 100 minutes for the midterm. Approximately how many questions are there? There are, you know what? I'll look it up because I'm not sure. I know there's 130 points, but I think maybe 50 questions would be my guess. But I will let you know specifics here.
I have a question about, all right, I have a question about, hello.
Hi, Brianna.
Hi. And so I have a question about the structures, like drawing of the structures and everything. Is that going to be more of a multiple choice? Are we freehanding, or how are we doing that as far as the different structures with the substrates and
Yeah. Okay. So just to wrap up the last one, there are 53 questions on the exam. For the structure stuff, because there is no way for me to get you to draw a structure and then upload it in the exam, there is a question that is similar to what you saw in the homework. So it's like identifying the right enzymes based on looking at the structure. But there is nothing where I'm asking you to actually draw out the chemical metabolites.
Okay. Thank you.
Yeah, sure. Yeah, so another question in the chat just came up. So like labeling the enzyme. Yeah, the enzyme names. So yeah, so like for it's the exact same instruction that I give on the homework. So it's literally label the enzymes. So I don't want you to be like, oh, it's CYP2D6, because that's too specific for me. I just need you to know it was a P450. Yeah, but labeling if it's UDP glucuronyl transferase, which the accepted labeling is UGT, that's fine.
Oh, I just have a quick question. G proteins can be coupled to adrenergic receptors and muscarinic receptors. Is that correct?
Yeah, that's a good question. So G protein coupled receptors as a family, there's like what are there? 50 of them, I think. There's 50 of them. I can't remember if it was 30 or 50. But they can couple to, the muscarinics are an example of G protein coupled receptors, just I think it's 1, 3, and 5. Beta adrenergic receptors, alpha adrenergic receptors are G protein coupled. Opioid receptors are G protein coupled. There's a whole slew of G protein coupled receptors. They're the largest drug target by far, by most drugs, because they control so much signaling. I don't know if you remember, there was a picture in one of our, maybe you guys don't have this lecture because I changed up how I delivered the material this year, but there's this like crazy slide that I have where it's like all of the different G protein coupled receptors, which G proteins they couple to, and then what their effects are. Because they pretty much control, I don't want to say everything, but like almost everything in the body.
So when we talk about activation, the majority of the activation that goes on the body is the G coupled receptor compared to like your voltage and your ligand. And so is it you have your ligand come in and then it activates the G protein receptor in the in the cellular, what do you call that? Cytoplasm?
Yeah.
Yeah. Okay. Gotcha. Okay. Because when I was looking at it, I was thinking just muscarinics were attached, but it can be a whole slew of them can be attached to G. Okay. Thank you.
Yeah. Keep questions coming. I'm going to see if I can find that slide to show you guys.
Mhm. But your M2 receptors are always in the heart, and your M3 are basically, I think the lecture was elsewhere. And your other ones are in the brain. So when you're looking at stimulation there, if you're not in the heart, if you're looking at your smooth muscle peripherally, it's your M3.
Believe so, yeah. So I got another question about nuclear activation. So the nuclear receptor activation, it's the only thing that's like different about nuclear receptor activation is the ligand itself needs to move through the cell membrane. And then once it gets through the cell membrane, it'll bind to a receptor, and that receptor will find a partner. And it can either be a homodimer, so like two of the same receptor, so like estrogen receptor to estrogen receptor, or it could be a heterodimer, so it would be, two different, it could be like estrogen receptor to, there's one that's just called RXR, which binds with almost all the other nuclear receptors. And so once you get the dimers bound, then they move together into the nucleus, and they'll bind to DNA and cause changes in transcription. And so some things about nuclear receptors, these are usually binding your steroids. The time that it takes for those to react is a little bit longer because you have to cause changes in gene transcription. And so that takes longer than something like an ion channel opening or closing, which happens in seconds or milliseconds. So nuclear receptors end up taking the longest to have an effect. What else is unique about them? The receptor signaling. While everything, like all cellular signaling, usually results in changes in gene transcription, nuclear receptors are the only ones that actually move into the nucleus, bind to DNA, and actually cause a change as soon as they can. So key points would be binding a ligand, in the cytoplasm, dimerizing, moving into the nucleus, and changing gene transcription.
We didn't really talk much about in the renin system. We just really briefly touched on the opposite side, the bradykinin side. How, I mean, is that something that we should also focus on, or?
Oh, I'm going to be terrible at that one because I actually, like, I never teach that section, so I'm not really sure.
Okay.
I think that whatever she covered in her video should probably be sufficient.
Okay.
Yeah. Sorry, I don't have a more specific answer for that.
Yeah.
So I found something that's similar to that slide that I had before about all the G protein coupled receptors. So let me show you what it.
Oh, that's cool.
So here you can kind of appreciate all the different things that'll activate a G protein coupled receptor. So they've got like your epinephrine, dopamine, dopamine receptors, certain histamine receptors, certain acetylcholine receptors, same for glutamate, calcium, GABA. So all of these things can interact through G protein coupled receptors. My favorite ones, or the ones that I think are most interesting, is the fact that like light can activate a G protein coupled receptor. So a lot of the ways that we see colors is actually through a G protein coupled receptor that gets activated by the actual wavelengths of light. So yeah, there's lots of G protein coupled receptors in the body.
And then do you have any tricks for, I hate to even use that word, but like when we see phosphokinase C, it's PKC, but then you might have an abbreviation for a protein kinase, something, and it's still PK. So I guess you just have to know where in the cycle that they show up versus being able to learn from their names.
Yeah, I don't even actually know where those naming schemes came from. My guess would be the order in which they were discovered, but I don't think it gets that specific in the exam.
Okay.
I don't like memorizing, so I try not to make people memorize things. If you've seen it enough from just studying and doing the homeworks and the journal articles, then you should know it well enough without needing to memorize.
Okay.
This is more not particularly preparing for an exam, but is there a way that once the exam is done, how do I put this? Is there a way that we could get like a feedback of what parts it seems like majority of the class is not getting, or a way for clarification for how the exam went?
Yeah, for sure. So I will probably, unfortunately, I can't just like release the exam. I will look into figuring out a way to provide some more specific feedback. The course is not cumulative, so whatever we do in the midterm won't be on the final, but I fully appreciate that you also just want to understand what you missed. So I will figure out a way, and you know, if individuals want that information, if you just email me, I will gladly go through it. If you want more specific information, I'll provide a breakdown of like how everybody did on each mod, not individually, but like as a group, how people did on each of the modules. But then, yeah, if you want that individual feedback, I can do that for you, for sure.
The other question that I have, in previous classes, what did you see to be the most difficult module for students?
That's a good question. I think people struggled with, on the Romets material, knowing the drug names. I think that was where I saw the most amount of struggling. It was a little bit hard for people to answer the cardiovascular questions, so like the module 3 questions, but I think that's because they ran out of time. So the people that had the time to get through it were okay. I think that if there was more time, I think everybody would have been okay. Personally, I find the next section to be more interesting. And so I think that the next section, like for the final, I think it's more fun and easy to study and more kind of black and white than these other drugs we've been learning.
Okay. I do have to go back to work, so
Okay.
thank you. I'll see you.
Yeah.
Bye, and good luck, everyone.
Good luck.
Thanks, Holly.
I think I'm done. I don't have anything else, so thank you very much.
Yeah, sure. Thank you so much for asking questions. I really appreciate it. Sometimes these things can be a little quiet and awkward, so. What about anybody else? Does anybody else?
I have one more question for her.
Sure, go ahead.
So, you know, on our nervous system, it talks about how a lot of these work through nicotinic receptors, but we didn't go a lot detail into the somatic. And it says somatic always acts on nicotinic receptors through acetylcholine. You know, and on our notes, it shows, you know, where it can come in, and the acetylcholine comes in and works on this nicotinic receptor and then goes to your effector organ. Where is the, sorry.
Oh, you're okay.
Where's the nicotinic working right here on the somatic? I know that they work on, what is that, muscarinic on the skeletal muscles, but where does nicotinic work on the somatic?
Ooh. Off the top of my head, I do not know.
Okay, because I know like where we had these synaptic, the pre and post, you know, your nicotinic receptor would be like right here, and then it would carry the signal onto your, you know, your muscarinic or your androgenic receptor. But it's just we didn't do a lot on the reflexes and stuff like that.
Yeah.
So I was just
your skeletal muscle there, when that acetylcholine is released, that is a nicotinic receptor there in your skeletal muscle.
Okay. I thought it was a muscarinic. It said N1. NMN1 is located in the skeletal muscle.
I think that NM is nicotinic muscular versus nicotinic neuro. So there's a little there's a little confusion about that. But I'm pretty sure the nicotinic is at that skeletal muscle, and that's nicotinic M for muscular.
Okay, so it's right at the skeletal muscle then.
Yeah.
Okay, because the other ones, you know, okay, gotcha.
Yeah.
Okay. All right, well, then that makes sense. It just goes directly to it.
Yeah.
Okay.
Okay, got a question on chat. Number 11 on homework 3 with respect to the pupils. What is the effect of the PNS and which receptor mediates the effect? I guess I'm confused. Okay, let's see. I'm pulling up 3, number 11. Okay. And if anybody also wants to chime in and help explain this, because this, again, this isn't really my material. I apologize, but so if somebody else also wants to jump in and take a shot at that.
Well, the parasympathetic nervous system is going to affect the radial muscle. That's the one that goes around the pupil. So when that is stimulated, that circular muscle is going to constrict, and that's going to make your pupil get smaller. So I'm not sure if that helps with the question. And then again, it's the M3.
Okay. Yeah, so I guess on this one, thank you. This one, it's, so, I mean, the difference between these, right, they're exact opposites. Right, so.
Yeah, well, if the sympathetic is stimulating the radial and the parasympathetic is stimulating the circular. So when the sympathetic stimulates your radial, it's going to contract, it's pulling back. So that's why you get the mydriasis, the dilation.
Mhm.
So it's weird because one system stimulates one muscle and the other system stimulates the other muscle.
That makes sense, yeah. I mean, it is weird, but it makes sense. Does that help, Ashley?
Well, I mean, I think you have to think about fight or flight, the fight or flight mechanism. And if you're thinking about sympathetic and parasympathetic, if a lion is chasing you, you're not going to want your pupils to dilate. You're wanting to get closer. To close, you're going to want them to dilate because you're wanting to be able to see. You're alarmed. You're ready to go. So on my notes, I actually have that as the opposite. I had the sympathetic nervous system causes the radial muscles to contract, which is going to dilate the eyes. And then you have the parasympathetic is going to contract the circular muscles, and then you're going to have the constriction of the eyes.
Can you say that again?
Yeah,
Because
I have the sympathetic nervous system. I have it right there. It's going to
stimulate the radial.
It's going to stimulate the radial, which you see that's dilation.
That's not dilation. Yes, it is. I'm sorry. That's dilation. But the radial muscle contracts,
causing dilation.
Yes. Mm-hmm.
And then you have your parasympathetic nervous system is going to contract your circular muscles, and then you're going to have it's going to constrict, causing your eye to look like that.
Yeah, that's right.
Mm-hmm.
Okay, I think she just has a confusion about the actual clear definition of meiosis and mydriasis. So just that, because she understands the receptors, it's just the clear definition which one is which. So I think that's where she's getting confusion as to which one causes the contraction and then which one causes the dilation.
Yeah.
Well, one trick is that if the word has a D in it, it's dilation, and that kind of makes it a little easier.
Yep. Super helpful, right? Yeah. The D is dilation. Thank you.
I guess you can think of it. Radial muscle has D, and then mydriasis has D, dilation.
Yeah.
Mm-hmm.
I was a little confused on the nice, what is it, the diagram of the eye, because I was really trying to figure out where the little points were on the eye, like where she was actually pointing on the structure for the eye. So it was a little bit like, okay, follow the line. Where is this actually pointing to?
Mm-hmm. Yeah, I think that's just hard. I mean, I think you can look things up on the internet and
Right, of course.
where she got this from. I know I struggle with some of those images as well.
Okay.
Which I'm glad you brought that up because I want to go through the exam and make sure that images show up because I know that had been a problem.
Okay.
So I'm glad you said something.
You're welcome. So I was like, I don't know where this line, I'm trying really hard to follow it. Because, it's so many layers to the eyes to where it's like, one muscle can be here and then another layer is a different muscle,
And we've got maybe 10 more minutes. So if anybody has anything, now would be the time. Here we got alpha 2, GI couple. Everybody in agreement? Alpha 2, couples 2.
Inhibition.
I don't know. I took something. These
I
computers.
would agree with that.
It is. Yeah. So alpha 2 couples to GI. And again, for things like that, if you're like in there and you're doing the research on it or you're prescribing drugs with it and you see it every day, you'll probably know that. But for the purposes of the exam, feel free to have a little cheat sheet in front of you where you've got what these are coupling to. That's something that like I would have known because I used to work on G protein coupled receptors all the time, but like haven't worked on them in a few years. So, those things kind of go away from the memory after a little while.
Mhm.
Where's everybody calling in from? Who's my furthest calling in from Fort Wayne?
East Coast, Maryland.
All right.
Oh, yeah,
I feel like Kristen might be Texas.
Oh.
Are you Texas? Where are you?
I'm in good old South Carolina.
All right.
Yep.
Do we have anybody further from that?
I'm in Dallas.
Okay.
Oh, all right. Valpo, Chicago. All right. Yeah, I think South Carolina still might. Oh no, Dallas would win. Yeah.
So y'all have snow up there?
Yes, we do.
Do you? How many inches?
Well, it's all kind of melted now, so it's only in the piles where we've shoveled it. But yeah, it snowed about 10 days ago. We got hit pretty hard.
Wow.
It's early for snow. It was early, so.
Bye, guys.
Yeah. All right. Well, let's wrap it up. And if anybody has any leftover, I'm going to post both the chat and the video if you want to look at it again, or people that couldn't participate want to look at it. And then if anybody has any questions, feel free to shoot me an email and I'll do my best to answer them, especially over the romance material. I do work with her. We share a wall, so it's not like I can't ask her if there's something that I'm a little confused on. So other than that, good luck on the midterm exam. And we'll see you guys soon, okay?
Bye-bye.
All right. Bye, everybody.